So continuation with the previous class where we have discussed about the uh, structure of the hemoglobin. So initially we discussed about the three important functions and three important properties of hemoglobin. Then I have already explained what is the cooperativity. Then we discussed about the structure of the heme and the structure of the globin. And when we discussed about the structure of the heme, we have already said that it has four pyrrole rings and four methylidine, four methyl groups and two propionic acid side chains, two vinyl groups. And the iron atom is present in the center of the 4-pyrin and it is present in Fe+2 state. And in this state, it can form 6 coordination bonds. 4 coordination bonds are satisfied by the 4 nitrogen atoms, and the 5th coordination site is satisfied by the imidazole of the histidine of the globin of, chain, and the 6th coordination site is meant for the oxygen binding. So I forgot to mention that in the deoxygenated hemoglobin, you can see here that the iron atom. Students, am I audible?
Yes, sir.
Yes, sir.
Yeah. In deoxygenated state, in deoxyhemoglobin, the iron atom is present 0.4 angstroms outside the 4 pi average plane of the 4 pyran ring. You can see here this is so when oxygen binds, you can see here upon oxygen binding, the iron atom is pulled into the plane, and of course the this movement in turn pulls the histidine residue that is in the fifth coordination site. As a result, the conformational change takes place in all the subunits. So this we will explain later. So therefore, in our last class, we have already said that the hemoglobin is present exist in two states. One is a deoxy state, another one is oxy state. And deoxystate is also known as the deoxygenated hemoglobin, or the oxystate is known as the oxyhemoglobin. Deoxystate is also called as the T state or tensed state or the tau state, and whereas the oxystate is known as the R state or the relaxed state. And as I already said that in a deoxygenated state, and all the subunits are stabilized by many ionic interactions between the salt bridges. You can, as I already said, that since all the subunits are stabilized by the inter-subunit interactions of electrons of ionic bonds, and these that's why the T state is also known as the tensed state. So coming to the when upon oxygen binding to the one subunit, and all these salt bridges would be progressively disrupted. As a result, the hemoglobin can efficiently bind oxygen, so in a cooperative manner. So this we will see later. And now coming to the, in order to understand the hemoglobin cooperativity, first we need to understand the hemoglobin oxygen dissociation curve. So the hemoglobin oxygen dissociation curve is nothing but it is the graphical representation of partial pressure of oxygen versus the hemoglobin O2 saturation. So in the, as we already said that in the lungs the partial pressure of oxygen is 100 tar, 100 tar. So that means when the hemoglobin moves to the lungs, and let us say this is the hemoglobin, and it is a tetramer, and tetramer, it has 4 oxygen binding sites, and in the lungs all the hemoglobin molecules are saturated with the oxygen. That means all the binding sites are occupied by oxygen. So that's what happens in case of in the lungs. So that means in the lungs, because of the high partial pressure of oxygen, or the hemoglobin is fully saturated, and that means the 98% of the binding site is saturated with oxygen. So 98% or the 100% means 100% means all the hemoglobin molecules are loaded with the oxygen, and they are carrying, each hemoglobin molecule carries uh, 4 oxygen molecules. So when hemoglobin moves to the tissues where the partial pressure of oxygen is 20 tar, 20 tar, again hemoglobin unloads its bound oxygen. So that means its saturation drops to its saturation, and where the partial pressure of oxygen is 20 tar, its saturation again drops to the below the 100%. That means the hemoglobin unloads its bound oxygen. So this oxygen dissociation curve is useful to understand cooperative behavior of the hemoglobin while when it is transporting oxygen, and we can also understand in what way the hemoglobin is a good cargo to transport oxygen, whereas why myoglobin or some other non-cooperative proteins are not good vehicles for oxygen transport. So here, what you need to understand is that the oxygen dissociation curve is nothing but the graphical representation of partial pressure of oxygen versus hemoglobin saturation. So 100% saturation means all the hemoglobin molecules are loaded with the oxygen. 50% saturation means only 50% of the hemoglobin molecules are saturated with the oxygen, and so on. So that means that is the saturation. The 0% saturation means 0% saturation means none of the hemoglobin molecules are carrying any oxygen. That means no oxygen is bound to the hemoglobin. So that is the meaning of the saturation percent. So 100% saturation means all the hemoglobin molecules are loaded with oxygen. 50% saturation means 50% of the hemoglobin molecules are bound to the oxygen, bound with oxygen, whereas the 0% saturation means none of the hemoglobin molecules are carrying oxygen. So now coming to the O2 dissociation curve for hemoglobin is sigmoidal. You can see here the O2 dissociation curve for the hemoglobin is it is a sigmoidal curve, S-shaped curve, whereas the myoglobin it is the hyperbolic curve. This we will tell you later, and why the difference. And coming to the partial pressure of oxygen is always represented for in order to understand the hemoglobin O2 binding affinity. We take a different we take here the P50 value, and P50 is nothing but the partial pressure of oxygen that is required to achieve 50% saturation. That means here, here, as I already said, that at 100 tar, 98% of the binding sites are occupied by the oxygen, whereas the at in order to achieve 50% saturation, how much of partial pressure is required? That is nothing but the P50. P50 means it is the oxygen tension that is required for the 50% saturation. So for in case of hemoglobin, it is 26 tar, whereas in case of myoglobin, it is only 2 tar. So that means it indicates that hemoglobin can achieve 50% saturation only at 26 tar or the 26 tar of the oxygen tension, whereas the myoglobin can achieve this same 50% saturation at a lower partial pressure. It indicates that myoglobin has the higher O2 affinity compared to the hemoglobin. And now coming to yeah, you can also see yeah, one more point also I forgot to mention in the last class. In deoxy state, in deoxyhemoglobin, as I already said that in deoxyhemoglobin, the iron atom is present 0.4 angstroms outside the plane. And in addition to this, in deoxygenated state, the deoxyhemoglobin also has 2,3 BPG pocket. This 2,3 BPG pocket. This is the 2,3 bisphosphoglycerate pocket. So this 2,3-BPG is a anionic molecule. It decreases the hemoglobin affinity towards oxygen by stabilizing the T state. So that means this T, the in only in T state this hemoglobin has the 2,3-BPG pocket. That's where the 2,3-BPG binds and stabilizes the T states. As a result, hemoglobin affinity towards oxygen reduces. So then now, as I already said that why hemoglobin is a good cargo to transport oxygen and compared to the other proteins, in order to understand that we need to understand the again O2 dissociation curve for the hemoglobin and the myoglobin and also the hypothetical non-cooperative protein. You can see in this oxygen dissociation curve. So we have this is the green line indicates for the myoglobin, whereas this one for the hemoglobin, and this blue line for the hypothetical non-cooperative protein. As we already said that hemoglobin is a cooperative protein in the sense of binding of one oxygen molecule and increases its further binding more efficiently to the other subunits. So this is exhibited by the hemoglobin, and and because of its tetrameric nature, the hemoglobin shows the cooperativity. And so when hemoglobin, in order to understand the importance of this cooperativity, so when you look at when we examine this O2 dissociation curve clearly here, so as we have already said that the partial pressure of oxygen in the lungs, in the partial pressure of oxygen in the lungs is 100 tar, whereas the partial pressure of oxygen in the tissues is 20 tar. So that means when it moves to the lungs, and it's 98% of the binding sites would be saturated, 98% of the binding sites would be saturated with the oxygen. That means its saturation level is 98. And when the hemoglobin moves to the tissues, and where the partial pressure of oxygen is 20 tar, and hemoglobin saturation drops to 32%. 32%. That means so its saturation drops to 32%. Therefore 98 minus 32, 98 minus 32 is equal to 66%. That means if you have confusion, once again I will draw here. You just see this is the hemoglobin dissociation curve. So in the lungs where the partial pressure of oxygen is 100 tar, 98% of the binding sites are occupied with oxygen. That is the hemoglobin. This is the hemoglobin O2 saturation percent. So that means 98% of the binding sites are occupied or loaded with oxygen. When hemoglobin moves to tissues where the partial pressure of oxygen is 20 tar, the hemoglobin saturation drops to 32%. Here it is 98%, and here the saturation drops to 32%. That means 98 minus 32 is equal to 66%. That means hemoglobin in the lungs, its saturation is 98. When it moves to tissues and its saturation drops to 32%, that means 66% of the bound oxygen is released near the tissues, but still it is holding the 32%. So that is the meaning of the saturation level here. So that means hemoglobin can release 66% of the bound oxygen when it moves from lungs to the tissues. And in the same way, you can see here the hypothetical non-cooperative protein. So in case of hypothetical non-cooperative protein, and near the lungs, the saturation is 63%. 63% of the binding sites are occupied by the oxygen. So when the In case of non-cooperative protein, where in the at a partial pressure of 20 tar, that is absorbed in the tissues, and its saturation drops to 25%, 25%. Therefore, 63 minus 25 is equal to 38%. 38%. That means hemoglobin can contribute 66% of its bound oxygen for unloading, and whereas a non-cooperative protein can only unload 38% of its bound oxygen. So that is the difference between the hemoglobin and other non-cooperative proteins. So from this we can understand that hemoglobin not only good in oxygen binding, it is also good in oxygen dumping or the oxygen releasing when it moves from the lungs to the tissues. Whereas in case of non-cooperative protein, it is not as good as hemoglobin in releasing the oxygen near the tissues where the partial pressure of oxygen is 20 tar, because non-cooperative protein can only unload 38% of its bound oxygen, whereas hemoglobin can unload 66% of its bound oxygen near the tissues. That's the hemoglobin is a good cargo for the O2 transport. And if you see the myoglobin can only unload only 7% of its bound oxygen to the tissues. So that's why the hemoglobin is the good vehicle for transporting oxygen from lungs to the tissues. And this is about so from this what we need to understand here, that the hemoglobin not only good in oxygen binding, and because of its cooperative behavior, it is also good in oxygen releasing when it needs to release in the tissues. So that's why compared to the non-cooperative protein, and hemoglobin is good in oxygen binding as well as oxygen transporting. And so coming to the so as I already said that when oxygen, you can see here, this is the porphyrin ring. As I already said that the iron atom is present 0.4 angstroms outside the plane. So upon oxygen binding in the fifth coordinate, sixth coordination site, and you can see here that the iron atom is pulled into the plane. As a result, this in turn pulls the histidine residue, and this histidine is the part of the globin chain. It could either alpha or the beta. As a result, the conformational change takes place. All the salt bridges, what we have seen in the deoxy state, would be progressively disrupted, and so on. So this conformational change is observed when upon oxygenation. And you can also see here oxygen binding the iron atom moves into the plane, and upon oxygenation, when iron atom moves into the plane, and the one pair of alpha beta subunits rotate with respect to the other pair 15 degrees. As a result, all the eight salt bridges that are present in the deoxygenated state would be progressively cleaved. As a result, the all the subunits can easily take up the oxygen. So that's what that's what takes place when upon oxygenation in the hemoglobin. So you can also see here that this is only the model to understand as we already said that the hemoglobin is a tetramer. It has 4 subunits, 2 alpha and 2 beta, and all these thin lines represent the salt bridges, and you can see here upon oxygen binding, all these salt bridges progressively becomes weak. As a result, they would be progressively disrupted upon binding of the oxygen. So that's what happens here. As I already said that when first oxygen molecule binds to the first subunit, and as I already said that the first conformational change that is observed here is that the iron atom moves into the plane, and as a result the proximal histidine residue is also pulled, and this proximal residue, as a result, this conformational change would be transmitted to the other subunits as well, because all these subunits are stabilized by the salt bridges and also other non-covalent interactions. and all these salt bridges would be progressively disrupted. As a result, all the subunits can, all the oxygen can easily bind to the other subunits as well. So this is the first conformational change that is observed when deoxyhemoglobin, when the oxygen binds to the deoxyhemoglobin and it converts into the oxyhemoglobin. Now, coming to this, we know that the hemoglobin not only transport oxygen, it can also transport carbon dioxide and protons from the tissues to the lungs for the exhalation. And so this diagram, you can see clearly that hemoglobin. So you can see here that when hemoglobin moves to the lungs, and where the partial pressure of oxygen is 100 tar, and it is hemoglobin the bound to the oxygen, and it forms the oxyhemoglobin. This oxyhemoglobin goes to the tissues, releases its oxygen, where in the tissues the partial pressure of oxygen is only 20 tar, and this released oxygen of course is taken up by the myoglobin in the muscle, and it stores in the form of oxymyoglobin. When the during the respiration, when the muscle during respiration, when the oxygen is required for the respiration, again the stored oxygen would be released for the aerobic respiration. And now, once the hemoglobin releases its oxygen, and there is this deoxyhemoglobin, and again uh, this deoxyhemoglobin again takes the protons and moves back to the lungs. So now we need to understand from where these protons are derived. So the rapidly metabolizing tissues produce carbon dioxide in the metabolism, aerobic metabolism, and this carbon dioxide, which in turn moves into the circulation, and it enters into the red blood cell, where this carbon dioxide is converted into the carbonic acid. You can also see here this carbon dioxide is converted into the carbonic acid, and this carbonic acid is further dissociates into bicarbonate as well as H plus by this reaction is catalyzed by the carbonic anhydrase. So the conversion of, that means the rapidly metabolizing tissues produce carbon dioxide. This carbon dioxide diffuses into the circulation, which in turn enters into the red blood cell, and this carbon dioxide undergoes hydration by the action of carbonic anhydrase and forms the carbonic acid. This carbonic acid further dissociate into the bicarbonate as well as H plus ion by the same enzyme, that is the carbonic anhydrase. And now this HCO3 minus and H plus ions, this HCO3 minus ion again moves out of the red blood cell from inside to outside in order to maintain the charge balance across the red blood cell membrane, and the one Cl minus ion enters into the inside. That means it is antipode. That means as HCO3 minus leaves the red blood cell, Cl minus enters into the red blood cell, and this is known as the chloride shift, or the Hamburger's phenomenon. And now this HCO3 minus has a higher solubility in the plasma, and this HCO3 minus again moves to the in the HCO3 minus is further transported in the lungs, transported in the circulation to the lungs. Near the lungs, again this HCO3 minus further re-enters into the red blood cell. Again the Cl minus again leaves the RBC again by the same process. Now this HCO3 minus further reacts with the H plus, again forms the carbon dioxide and H2O by the same enzyme carbonic anhydrase, and this CO2 would be exhaled during the respiration. So that's what happens in case of the transport of oxygen and carbon dioxide by the hemoglobin. And now we know that the carbon dioxide, the hemoglobin is loaded with the oxygen in the lungs, and it is and the oxyhemoglobin moves to the tissues and releases this one. And as I already said, that hemoglobin, the H plus ions are also carried by the hemoglobin and to the region of the lungs. So that's why, as I said, that hemoglobin not only transport oxygen and the carbon dioxide, it also regulates the pH of the blood. And now coming to the once the hemoglobin releases the oxygen at the tissues, as I already said that it also transport carbon dioxide, and this carbon dioxide is transported back to the lungs from the tissues, mainly in two ways. And 15% of the carbon dioxide that is produced is transported in the form of carbamate, and the 85% of the carbon dioxide is transported in the form of the bicarbonate. As I already said, that bicarbonate is the product that is formed when the carbon dioxide hydrated with the carbonic anhydrase enzyme. And you can also see here the carbon dioxide that is produced in the tissues also uh, react with the amino terminal nitrogen of the polypeptides of the hemoglobin. As a result, the carbamate is formed, and this carbamate, 15% of the carbon dioxide is transported in this form, that is the carbamate form. And whereas the 85, most of the carbon dioxide, that is the 85% of the carbon dioxide, is transported only in the form of the bicarbonate. So this is and of course very minute amount would be transported in the gaseous form from the lungs to the tissue. So this is about the carbon dioxide transport. So now coming to the Bohr effect. So this Bohr effect is first invented by Danish scientist Christian Bohr, and this Bohr effect indicates that when the pH decreases, when hemoglobin moves from lungs to tissues, and the pH drops from 7.4 to 7.3. In the sense, in the lungs, the pH is 7.4, whereas in the tissues the pH is 7.2. So when hemoglobin moves from lungs to the tissues and there is a change in the pH, and so this reduced pH in turn decreases the hemoglobin affinity for the oxygen. As a result, the hemoglobin has to unload its bound oxygen. So this phenomenon is known as the Bohr effect. And so you can also see here that once again you can see here that, as I already said, that so the rapidly metabolizing tissues produce carbon dioxide, and this carbon dioxide, by the action of carbonic anhydrase, forms the carbonic acid. This carbonic acid in turn further dissociates into bicarbonate as well as the H plus ions. These H plus ions are responsible for lowered pH near the tissues, that is 7.2, whereas in near the lungs the pH is 7.4. So that means these H plus ions are responsible, and these H plus ions, again you can see here, this lowered pH causes the hemoglobin affinity towards oxygen reduces. As a result, hemoglobin unloads more oxygen near the tissues. So if you look at the molecular basis for this effect, you can see here that, as I already said, that in deoxygenated state, all the four subunits are stabilized by 8 salt bridges, and upon oxygenation, all the salt bridges would be disrupted. And when hemoglobin so as a result, when the hemoglobin in the R state, when it moves to the near the tissues, again, because the lower partial because the pH is lower compared to the 7.4, the pH is 7.2 near the tissues. Therefore, that means these H plus ions that are coming from the carbonic acid dissociation in turn causes the proteination of the histidine 146. We can also see here. So this proteination of this histidine 146, which in turn facilitates the formation of the salt bridge with the aspartic acid, that is negatively charged aspartic acid residue. As a result, hemoglobin switches from the R state to the T state. As a result, when hemoglobin is moving from the R state to the T state, hemoglobin has to unload its bound oxygen. That's why. So when hemoglobin moves from the lungs to the tissues, there is a change in the pH, that is from 7.4 to 7.2, and this lowered pH means the higher H plus ion concentration in turn protonates the histidine 146 present in the beta chain, which is involved in the salt bridge formation with the aspartic acid. As a result, the R state becomes a T state. Therefore, the hemoglobin in the T state, the hemoglobin has the lower affinity to oxygen. Therefore, it has to unload its bound oxygen. So this is observed by Christian uh, Bohr in 1904, and this phenomenon is known as the Bohr effect. You can also see here that when the pH so when the pH is reduced from 7.4 to 7.2, here you can see the two dissociation curves, and when the pH is lowered, the O2 dissociation curve moved towards the left. So that means if it is moved towards left means it has lower affinity. If it is moving towards right means it has a higher affinity towards oxygen. So this you can clearly understand that. For example, see in this case, in order to see the 50% saturation, in order to see the 50% saturation, 50% saturation at lower pH, the higher oxygen tension is required to achieve the 50% saturation, whereas at higher pH, lower oxygen tension is required to see the same 50% saturation. That means that the hemoglobin affinity towards oxygen reduces as the pH reduces from 7.4, that is from 7.4 to 7.2. So this is known as the Bohr effect. And now coming to, there are many effector molecules regulate the hemoglobin affinity towards the oxygen. Another molecule that influences the hemoglobin affinity towards oxygen is the 2,3-bisphosphoglycerate. So this 2,3-bisphosphoglycerate 2,3-bisphosphoglycerate is the anionic molecule, and you can also see here this is the glycolysis, and we know that 1,3-bisphosphoglycerate is the intermediate, and the 2,3-bisphosphoglycerate is formed from the 1,3-bisphosphoglycerate by bisphosphoglycerate emitase, and this takes place only when there is a low levels of oxygen. The red blood cell synthesizes the 2,3-BPG, and here you can see that when the hemoglobin moves from the lungs to the tissues, in the lungs the partial pressure of oxygen is 100 tar, whereas in the tissue the partial pressure of oxygen is only 20 tar. So when hemoglobin moves from the higher partial pressure to lower partial pressure, so because of the lower partial pressure, so in the glycolysis, instead of continuing here, so instead of continuing from glucose to pyruvate, the 1,3-bisphosphoglycerate also is converted into the 2,3-bisphosphoglycerate. As I already said that when when there is a sufficient oxygen, glycolysis would continue. When there is the oxygen tension is less, the 1,3-bisphosphoglycerate would form the 2,3-bisphosphoglycerate, and this 2,3-bisphosphoglycerate, you can see here it is a anionic molecule, and this 2,3-bisphosphoglycerate binds to the 2,3-BPG binding pocket in the deoxygenated state. As a result, 2,3-bisphosphoglycerate stabilizes the T state. Therefore, the 2,3-bisphosphoglycerate reduces the hemoglobin affinity towards oxygen. So that means when and you can also see here, since it is an anionic 2,3-bisphosphoglycerate is an anionic molecule, and it interacts with the positively charged amino acids lysine, histidine present in the histidine 2 and histidine 143 and lysine 82 present between the two beta subunits. In the sense, this 2,3-BPG pocket is present between the two beta subunits, and since it is a negatively charged molecule and it interacts with the positively charged amino acids, mainly histidine 2, histidine 143 and lysine 82, as a result it makes a cross link between the two beta subunits, and this you can see here 2 beta subunit. As a result, the T state is stabilized. Once the T state is stabilized, obviously the hemoglobin has to unload its bound oxygen, because in the T state hemoglobin has the lower affinity for the oxygen. So this is about the this one. So coming to the carbon dioxide, as I already said that one point I forgot to mention, as I already said that in the when hemoglobin is carrying carbon dioxide, 15% of the carbon dioxide is transported in the form of the carbamate. Only remaining 85% is transported in the form of bicarbonate. When the carbon dioxide when the carbon dioxide forms a carbamate with the amino terminal of the hemoglobin, and the negative charge is formed, and this negative charge in turn facilitates the formation of salt bridges. As a result, the T state is favored. Therefore, the hemoglobin affinity towards oxygen also decreases. Therefore, with increasing the carbon dioxide concentration, the O2 dissociation curve moves towards the right. That means with increasing the carbon dioxide concentration, the hemoglobin affinity towards oxygen again reduces. And and in addition to this, in addition to the pH and 2,3-BPG and the carbon dioxide, there are many other allosteric effectors also influence the hemoglobin affinity towards oxygen. These are you can see here that the temperature also can influence the 2,3 the affinity of the hemoglobin towards oxygen. So now, as I already said that when the as I already said that we have already seen that when the hemoglobin, as we have already said that, so with increasing with increasing the carbon dioxide tension, the O2 dissociation curve moves towards right. With increasing 2,3-BPG, the O2 dissociation curve moves right, and with increasing H plus ion concentration, that means reducing pH also shifts the O2 dissociation curve towards right. And you can also see here the increased pH, decreased 2,3-BPG, decreased partial pressure of carbon dioxide concentration shifts the O2 dissociation curve towards the left. And last one is the temperature. So with increasing temperature, the basal metabolic rate also increases in the red blood cell, which in turn increases the synthesis of more 2,3-BPG. As a result, the O2 dissociation curve also towards the right. So when the O2 dissociation curve shifts towards the right, it means that hemoglobin affinity towards oxygen decreases. Whereas when the O2 dissociation curve moves towards left, hemoglobin affinity towards oxygen increases. So this is the so that means the increased temperature, increased carbon dioxide concentration, increased 2,3-BPG, increased H plus ion concentration increases the hemoglobin affinity towards oxygen. So when the hemoglobin decreases the hemoglobin affinity towards the oxygen. So that means all these parameters like temperature, carbon dioxide, 2,3-BPG, and the pH, decreased pH, all these decreases the hemoglobin affinity towards oxygen. As a result, the P50 value increases. As I already said that you can see here that the P50 is the oxygen tension that is required for the 50% saturation. For the 50% saturation. So when the affinity is reduced, the P50 value increases. That is that you can that you can understand from the diagram. So this is about the hemoglobin, the uh, affinity towards oxygen, and the effect of 2,3-BPG, carbon dioxide, and the H plus ion concentration, and also the temperature. So now this is about the so now uh, we have one more small topic, that is the different types of hemoglobins, and I think this we will take it in next class.